your research, and I also want to present you with our research, but not only that, but also to give you some details about what we think in our field of studies, so computational neuroscience, are the pressing questions, and not only that, but to give you an idea of the approach we want to take to tackle these questions. Now, Mihai sends his regards. He would have really liked to be here, but he can't, unfortunately, and I'm taking his place for the talk. Now, here you see our group logo, and I'll refer to this quite some times during the talk, because it captures some aspects of what we think are these pressing questions. I want to start with this general introduction. So the title is From Biology to the Silicon Substrates, and this drives us. We want to understand what biology does. We humans are incredible inference machines. So we can recognize faces, persons, abstract concepts. We can theorize about them, all while having a very little energy budget. We've heard of this quite a few times today. And we want to understand this as a group, what's driving this efficiency, and understand the parts that are relevant for this efficiency, to be able not to only understand it, but then to build something out of this. So silicon substrates, new computing devices that make use of this, of our understanding of the brain. Now, I want to begin with a few ideas. If you start with computational neuroscience, and so von Neumann did this apparently, like Wolfram said this morning, and if you have a physicist's perspective, then you might think of the brain as being a big machine made of small parts, and it's very easy to distinguish what each small part of this machine contributes to the whole thing. So there's a close association between the small moving parts and the big purpose, this inference. Now, if you talk with your experimental colleagues, then it turns out it's rather messy. So this is a more apt representation. If you look at first sight, then what's going on in the brain is it's really complicated, it's noisy, it's not clear what's going on. Now, this is obviously also very tongue-in-cheek. If we look at different levels, we understand very well what's going on. But there's no clear understanding of how these things come together. And this is one of the big questions that we have. So understand it well enough to be able to build it. This is a definition of understanding that was put into words by Richard Feynman. What I cannot create, I do not understand. So we want to identify the relevant parts of the brain, of our neuronal system, to be able to build it. This is, for us, theoreticians, reminded that we shouldn't only write down theories, but we should test them. We should build something out of it, create simulations or even better devices that work like this. Now, I've said, and we've heard about this already, that we know on a very small level, we know what's going on in the brain of these ion channels and all those things. And we could just put this together and create a big simulation. Now we have lots and lots of neurons, so this would be a very huge simulation. And we can compare this to other kinds of simulations, for example, cosmology simulations. And if you want to simulate the universe, it turns out in a comparison, again speaking tongue-in-cheek, this is rather simple, because the particles that we have in the universe, they're largely identical. We have very good abstractions of their interactions. They're relatively simple and short-ranged. And we have a lot of techniques to deal with this simulation. So there's a lot of number of particles, or higher number of particles, so we can use thermodynamic limit, or we can use mean field approaches. This does not hold for the brain. Then we have highly diverse particles, or neurons. We know this from biology. And the interactions of these neurons are very, very complex. They're time-dependent, time-delayed, long-range, short-range, all these kinds of things. So it's very complicated what to do. And here you see the group logo again. We have to take a step back. We shouldn't include all of the biology that we know, all the level of detail. We have to make some kind of abstraction, so identify what could be the relevant biology and use this to build then on this abstraction level models. And use these models to see whether our abstraction level is actually correct. Now, I thought about making an example of what we can abstract away, but I didn't want to disturb anyone, and there's so many different things and there's so many different opinions where people might think that some things are relevant while others don't think they're relevant, and they could just be abstracted away. Now, if you have one abstract model of neurons, what we can do again is try to simulate this. Now, we can put this on a huge computer and try to see what's going on. The human brain has 100 billion neurons approximately, so it would be a very huge simulation. For this, we need some scaling. Now, if we distribute the computation, the simulation over different compute nodes, this will still scale not nicely, because the communication between the different nodes, compute nodes, will slow down the process. But not only the scaling is a problem. What's another problem is that the simulations themselves are very much slower. Now, how much slower depends on the actual level of detail that you look at. But you can generally say it is slower, and most of the time orders of magnitudes. This means you can simulate simple processes, simple time dynamics very easily, and even simple learning tasks in these abstracted neurons. But for more complex learning tasks, or even simulations of development, this is not feasible to do in simulations. So we have to think of something else. Now, the devices here are general purpose compute powers. And the next thing to do is obviously not to use these general purpose compute devices, where you could also do the cosmology simulations, but come up with something more specific. We've already heard the term neuromorphic hardware today. So hardware which is designed specifically, so it's a dedicated purpose to emulate or simulate devices. What you do here is, again, look at biology, abstract something away. So this is the Hodgkin-Huxley model there. And then one particular way, if you want to do physical computing, emulation, is to build a model which behaves like the one that we're interested in. Here, what people have been doing, and you see this on the right side, this is neuromorphic hardware, analog neuromorphic hardware developed by the Electronic Visions group in Heidelberg, where they build circuits which behave like neurons. So the dynamics of these circuits are very similar to the ones that we see in the neurons. And if you have these circuits and put them in a chip, then you get these nice-looking chips, and the voltages on these chips, they actually correspond to the voltages that you have in a neuron. And this is, another part of our interest. If you have these chips, then it's not only it's cool to have them, but it's even cooler to make them solve some task. So change the parameters of these chips, of these networks that you put on these chips. And this is where the whole group logo comes together. So from the biology that we think is relevant, we want to build some hardware which is efficient in simulating or emulating this part. And we want to devise algorithms which can effectively train these tasks and these hardware. Now, this is the general introduction, and I want to go into the more specific research. I want to start with credit assignment in cortical hierarchies, and more specifically in the visual cortex. Now, maybe the biologists shouldn't listen too closely, but this is a very abstract view, obviously. But what happens in the visual cortex is we get from the retinas, from our eyes, we get raw visual data that is going through hierarchical layers. And in these hierarchical layers, there's some edge detection, some pattern detection going on in some sense. And then there's abstract concepts on the top. Now, these examples, these two actresses and the building are very, very peculiar. They're taken from an actual study where subjects were tested and given images of these actresses or these buildings, and they found neurons in the study that were active for these channel concepts. So different pictures of the actresses or the building, or also just the writing, the name of this building, for example. And they found neurons which were active for these particular concepts. Now, this is biology, again, or some abstraction of biology, but it serves as the motivation for artificial neural networks, which started with using visual data in a hierarchical fashion with some units which do some computation in order to generate some abstract concepts, for example, image classification. If you do this in an ANN, if you're just interested in getting some classification accuracy, you can define, or usually define a cost function which measures how good you currently are, and then do gradient descent, so update the parameters given the derivative of this cost function with regards to the parameters. Now, if you do this in an ANN, then you can just assess all information. You don't care about locality or anything else, robustness to noise. You have digital computations usually. If you want to say something about biology or implement this efficiently in some hardware, then those things get interesting. So physical limitations, like locality in time or locality in space for these update rules that you guess, and also the robustness to noise. And this is a very general topic where we're interested in. And one specific way of thinking about this is using energy-based approaches. Now, as I said, I'm a physicist. So thinking in terms of energy is very natural for me and also my other physicist colleagues in the lab. But this is not just our way of thinking about this. This is what a lot of people do. So equilibrium propagation, if you know this, or other approaches also deal or are formulated in this energy-based approach. And what you do there is you formulate some energy that you want to minimate. So physical systems try for a state of low energy. And in this energy, this is twofold. One captures the dynamics of the network, and one captures this cost again, or here it's called loss. So the difference between the current state, R out, and the target state, R star. And from this energy, you derive the network dynamics, and you also get some kind of parameter update based on a gradient of some sort. And what you get is you have a system. So in this energy, or in these two equations, there's two parameters: beta, which measures whether you're given a target or not, and the learning rate eta. Now, if you set eta 0, then the parameters don't change, so the network just evolves in time. If you give some constraint, some external constraints, so some target or some reward, then if you don't change the parameters, you might have it, if you present some sample which has not been viewed by the network, that you increase the energy. This is an unfavorable state. We don't want to have this. So we turn on learning, we increase the learning rate, and then go to states of low energy again. And if this is then fully implemented, then we have a system which learns based on input, changes its state, its parameters, given some input. One particular example that I want to show you is from a paper called Latent Equilibrium, Fast Computation with Slow Neurons. So now we're getting into the actual details. Why slow neurons? Usually neurons do some low-pass filtering of their input. Here on top in this violet line, you see the voltage, which is the dynamic variable of a neuron, given some input. First, the input stays constant, so the output of this neuron stays constant. Then the input suddenly changes. And you see this by this dotted line. This dotted line is also the target for this neuron, what this neuron should reach. Now, once this input changes, the neuron doesn't abruptly change its state, but it has this exponential decay to the new resting state. In this setting here, this is a very common, so leaky integrator neuron, commonly used neuron type in simulations, in theories. Here, the learning rate is set to 0, so there's no learning going on. In the bottom panel, you see the weights staying constant the entire time. Now, if we have a learning task and we increase the learning rate, we usually would depend the learning on the difference between the target and the current state. Now here, because of this delay in the input, we have some offset, and this would be used to change the parameters. So we get some change in the weights given this difference. Now this is not something we actually would want. We would want, so the voltage here approaches this target value in the end, but because we have some learning going on, and we change the weights, and we also see that at the very end here, the values of the weights are different from when they were before. So we have unlearned something. this is not what we want. This is the problem slide, basically. For this problem, there's a few known solutions. We can turn down the learning rate, which makes the training insanely long, often infeasible, or we can phase the plasticity. So we can tell the network there's a settling phase, don't learn anything here and only start learning here. This is also not a really nice solution. We came up with a better solution, and this is using what we call the perspective voltage U brev. So this is this one here, which depends not only on the current state of the voltage, but also on the time derivative of the voltage. And if this U brev determines the output of the neuron, and thus also the difference between the target, then this neuron does not unlearn anything, but the information travels basically instantaneous through this network. And this instantaneity is still there, even though the network itself, or the neuron itself, is slow. Now, this is the concept. We've used this with these slow neurons on a few benchmark tasks, so the typical MNIST cipher, and also some particle collision dataset Higgs, and reach state-of-the-art accuracies with this. So this performs really well. It does what you expect it to do, so backprop in this in the setting with very fast simulation times, very short simulation times. And I should say here that this perspective voltage, this is actually something that you can observe, or there's some studies that observe this in actual biological neurons. So this is not just a purely mathematical trick, but it looks like this can happen in actual neurons as well. Now, what we can combine this with is not only have these neurons in this ANNs, or not ANNs, in rate-based neurons, but combine it with a circuitry which is biologically biologically plausible. So this is based on the Sacramento model, where you have very complicated neuron types. I will not go into detail, but the connections here are biologically plausible, and they shape what you can observe somewhere in the cortex. And you can then simulate these more biologically plausible models. And why the reason you can do this is having this new neuron type which has this perspective firing. Only with this you can actually, it's feasible to do these simulations. This was all published in last year's NeurIPS. And it's done for using classification tasks. We can also look for biology if we want to do more than classification, so if we want to do generation. Now, this is a nice paper by Nicolas Desprois and Jakob Jordan, which looked into using the concept of dreaming to increase structure in latent spaces. This is unsupervised learning for most of the time, where I'll just go very briefly through this, where we have a forward pathway, the green arrow here, which creates from some input, some latent space, and from this latent space again creates some picture of the visual input. And it compares those two, so something like an autoencoder, but with different phases, those two phases here. These resemble what we see in dreaming, where we both have one phase, the non-REM phase, where we replay memory from this latent space to create visual input, which is then trained or used for the forward pathway again, and also a REM phase where we not only use one input from the latent space, but multiple, combine them, so have some actual creation, some dreaming up of new images. And this method, which is shown here, it helps shape the latent space, and this latent space can then be used, for example, for classification, for better generation, all these things. The neural model that I've showed here is, the last one was an actual artificial neural network, where just the training method is inspired a lot by biology. Also, if this is vaguely familiar and you think this is something related to GANs, general adversarial networks, this is true, but it's, or not but, and it's an biological implementation or an idea for biological implementation which has experimentally testable hypotheses how the learning is going on during streaming in the brain. The neuron model here are rate-based neurons, so neurons which communicate at all times with a floating point number, and they have no structure. If we look at the brain again, we know since a long time that neurons are more complicated. So neurons have first some structure, so there's a difference between point neurons and compartmental neurons, and also the communication is not continuous in time but event-based. Here you see a voltage trace of a neuron. You see this voltage, this is a dynamic variable over time, and it changes continuously, but it has these spikes. These are the action potentials, the discrete points in time where this neuron communicates with its after partners. And only at these points it communicates. From these spikes you can get a spike train, which is often called S of t. And this is a sparse communication, which is one idea where the energy efficiency in the brain comes from the sparse communication. You can go back to a rate actually, so not all rate-based neurons models are inherently unplausible biologically, but if you want to look into more detail, then looking at the spikes is really important. For this, I want to give some more details on actual neuron models. Here you see an experimental setup of circles representing neurons, where we probe one neuron, the voltage of one neuron, when it receives some input chain from two neurons chain. Now you see that at the beginning, if there's no input to the neuron, then it just stays with its voltage around some resting potential, also sometimes called leak potential. And if it receives this input, then the voltage increases. Again, this is this low-pass filtering, so slow increase. And if there's no more input, then it decays back to the leak again. If there's enough input, so multiple input spikes, then it might cross the threshold, and if it does, then this output spike is generated. This is a very generic view of what's going on in neurons. A very specific model is the Hodgkin-Huxley model, which has been around some time, which models this very precisely on the level of ion channels. An abstraction of this that I want to give more detail about is the leaky integrated fire model, which you see here, the differential equation and the equivalent circuit. Now, if there's no input for this model, for this neuron, so ISIN is 0, then this just says that the voltage, U, decays over time back to the leakage. So this just gives you the difference from the leak, and the voltage will decay back to this leak. If there is some input, then this is slowly integrated. And this is a very often used neuron model for simulations. 2 points I want to make about this is if you give some constant current to one of these neurons, what you get is a response curve, so the rate of a neuron which gets this input. So on the left you see this constant input. What you get is basically rectifying linear unit shape, with a little bit of an offset. If you give some Poisson input spike input, so stochastic input, what you get is a logistic or sigmoid function. So if the number of spikes is too little, or there's too less excitation, there will be no output. And if you have enough input, then you'll get some maximum output rate. Now, I've started talking about stochastics, and this is the introduction to the next part, learning in stochastic models. Now, Damien already talked about stochastics, so this Monte Carlo or Bayesian view of the brain. I want to give an illusion as well for showing why this could be the case, or why this is a good hypothesis for the brain. So this is an illusion which, if you look at it, looks to you either as a duck or a rabbit. It will very unlikely look like both at the same time. It's either the one or the other. This is one reason for the sampling hypothesis, so that our brain is actually sampling from some probability. Another example here is this image, this illusion, where you either see a forest with some trees in there, or you can see a taiga. This is again a like hint why this could be implemented in the brain, because if you walk through the forest actually, and if you see a taiga, it's very important for you to actually recognize this taiga and then run away and not see just the trees in the forest. Now, if you want to deal with an actual statistical model, so with probabilities, how we often write this down is probabilities of some state set in this form. This 1 over set is the partition function, which deals as a normalization factor. Now, if you have a state space of an interesting problem, then the set is way too large to compute. There's too many states to calculate. So we have to find some other way. And one of these ways is sampling. So to get random numbers, basically a random state set, and of these sets, they should be of the states which have the highest probability, so that their occurrences in this state in the set resembles the probability. I'll not go into the details about sampling, but it's a method to deal with huge state spaces in an efficient manner. Now, here you see one of this schematic of this. If you have a state space here, two-dimensional, with two places of high probability, so this two red dots, then you want to draw samples for the sampling which are close to these points of highest probability in order to get this correct or a good set, which you can then use for the computation. Now, you see that there's lines drawn between those points. And this is because in usual sampling, what we do is, starting from one sample, we want to produce the next one. And we do this with some transfer function from one sample to the next one. If we do it in Gibbs sampling, we get a logistic function or the sigmoid function. And this is what I've just showed you is the case in a neuron. So if you, the output rate, if you look at very short times, is just the probability that an output spike will occur. So this activation function of a one neuron is very closely related to this transfer function between samples if you do some sampling. And you can exploit this in a very nice way. So if you have set up a network of neurons, you can look at it at any given time. And at this dotted line, or dashed line, you can see that one neuron has just fired, so it's in a refractory state. So you assign this the vector, the state 1. The other neuron, it has not fired in the recent time, so you assign this the state 0. Now, because the way the neurons interact with each other is via the spikes. So if one neuron has just fired, so it's in state 1, then it will have an impact on the surrounding neurons, weighted by these synaptic connections. And this makes this connection very, very obvious, and you can use this to then actually do sampling in spike-based systems. You train this with contrastive Happian learning, so looking at the correlations of these units between the biased and the non-biased model, and then can use this for discriminative tasks or also for generative tasks. So on the right, you, for example, see a network which was trained on MNIST classification, and it just dreams up new numbers. And if you look at it, most of the time you can recognize what kind of number this is. So this is a generational or an example of a network that samples new images. Another example application of this is the following. This has been done with our colleagues in the institute in Heidelberg, so the Kirchhoff Institute for Physics, and it's based around the idea that if you have a many-body state quantum system, then you need exponentially many parameters to describe this. Our colleagues, the quantum physicists, came up with a nice way, so the positive operator valued metric norm that gives you a connection between this complicated function with something that we know, just a probability times a function. And this is something we can sample again from, or we can train a network to sample the correct probability distribution and then check for specific observables. One of the observables that we tested is the bell, or one bell variable, which in this system tells you whether the system that you're looking at is in a quantum or in a classical state. And we can resemble in this spiking system the state well enough to be able to show that this quantum system that it's based on violates this classical bound. So this violation of this bell bound is a very cool thing in quantum physics, and we showed that we can do this in a LIF sampling system. And it's not only just a LIF sampling simulation, it's actually an emulation. So this has been run on neuromorphic hardware. And this is where I want to go now. We've heard already today a few things about the use of physical computation. I want to give one example again, which is very close to one that Wolfram showed. The comparison between how we can do a multiplication digitally or in an analog fashion. On the left, you see a 3-bit multiplier in a digital fashion, and you see that this is a very complicated circuit. It takes two 3-bit numbers and outputs a 6-bit number. Now, if you want to do this in an analog fashion, in an electrical circuit, what you can just do is use a resistance pumped from some current through it, and if you observe the voltage, then you get the multiplication of those two numbers. Now, this is a schematic which should tell you that it's using analog devices can be, or analog computation can be very simple, simplifying the computation, but it's just a schematic. There's other problems. So on the left, you get a bitwise correct computation. On the right, you might have noise, you might have changes in temperature, which corrupt your result. You get it fast, but it might not be as nice as the full bit one. But what you do, if you want to create with this idea of analog circuits, analog physical computation, new circuits, is you get some biology, some relevant abstraction of biology, put this in an equivalent circuit, so this is this equivalent circuit of the LIF neuron, and then design circuits which you put on device. Now, the circuits that you see on the left are actually not the one that is implemented on the right, but it's the ones, oh sorry, this was bad. It's the one that we used to create some very small example machines, which are just for outreach purposes, but it's an actual analog computing device. So it's an actual analog LIF neuron which is implemented here. With multiple, so a couple of hundreds of these, you can connect them, play around with them, and actually make them solve some very, very simple tasks. What the team in Heidelberg did was do this in a more scientific manner. So put a lot more circuits on one chip, do it in an integrated fashion. And now they have, so the electronic visions group in Heidelberg, they've got the BrainScales 2 system, which is a mixed signal neuromorphic computer, neuromorphic hardware, where you have these voltages available on the chip. So it's it's a physical computing device. If you prepare it in a correct fashion, you just have to provide some input and then can watch what's going on there and create, get the relevant information out of this. You don't have to solve equations at each point in time. You don't have to time step through the dynamics. You just watch what's going on and get results out really, really fast. It's actually sped up compared to biology, so relevant time constants are not like in biology on the order of milliseconds, but on the order of microseconds. What you can do on these chips is actually look at some very simple biological simulation or emulations. So on the left, because there's the ADEX neuron model implemented, it's a bit more complicated than the LIF neuron. You can represent or reproduce spiking patterns in these neurons. So this is the NOT spiking patterns which are done on the hardware. On the right, you see a small network of neurons which solve a navigation task which replicates navigation in a bee brain. So a bee which starts in a nest, goes out to find some flower, and then has the task to return to the nest in a very straightforward fashion. And this is done on the chip. And yes, you can run these emulations there. The last topic I want to talk about is a very specific one on this chip. If it's about learning with the actual spike times on this chip. I've shown you something like this already. So it's the voltage dynamics of a neuron which receives some input. So the voltage is constant until it reaches, gets the input, and then it reaches the threshold and it's reset at this point. This is the way the live dynamics look like. And for a long time, when people wanted to optimize this, they thought that this reset that happens at the output spike time is non-differentiable and makes it really hard to learn to do some optimization because it's usually based on some gradient that you want to do. What we did in our work is to change the question from did this neuron spike, so did this reset happen, but when did it happen? Because the spike time, so the point on the axis of this output spike, this is a floating point value. It's actually a differentiable function in specific cases that we found. And this differentiable function, you can calculate the derivatives. So there's the two derivatives here. The first one is the derivative of the output spike time with regard to the input weights. So what happens if you change the weights with the output spike time? And the second one is the derivative of the output spike time with regards to the input spike time. So if you change the input spike times, how does it change the output spike time? And what this gives you is the power to connect a difference in an output spike time of some hierarchical network with the input, with the spike times and the weights in a hierarchical network. So you can do gradient descent in this network of spiking neurons and solve, for example, image classification tasks. We did this on the MNIST dataset. You just see the training progress over time, you see the confusion matrix, and on the right you see samples of the voltages which the chip gives us. You actually see multiple repetitions of each test, and you see that here these curves are not on top of each other, and this is a result of the asynchronous and the analog nature of this BrainScales 2 hardware that we're using. The results we got accuracy-wise are quite good, and also for the actual energy efficiency. So the whole duration for this test of this 10,000 images took going from Python code to the C++, which gives some program to the FPGA, executes this on the ASIC, and then the whole way back. This took less than 1 second. The power consumption of this ASIC in this time is approximately 200 milliwatts. So this comes down to an energy per classification of 8.4 microjoule. Now, we've already heard about memristus, so it's, I think it was actually the same order of magnitude that Damien just gave. For an electronic device, this is really, really cool result. And for me, it was really, this power consumption was just a number until we switched chip iterations. So when we went from the first iteration of BrainScales 2 to the second one, we somehow measured a power consumption which was 15% higher, so 1/6 higher. And we went to the lab and tested quite a few things until we realized that there were some diodes still on the chip which consumed 1/6 of the power that was actually needed for the total consumption. This gave me a feeling for the actual power efficiency of the system. With this, I want to conclude my talk, and I want to stress what other people have said again. So if you want efficiency for computation, we need parallelization and in-memory computing. We have one device, one system where we know that this works. This is the human brain. So currently it's a very good idea to look at this to understand what's going on and to build systems which are shaped like this. And with this, I want to thank Mihai, my supervisor, again, and the whole group, actually multiple groups. So thanks for your attention. Yes?
Yeah, thank you for the nice talk. You also need to model sort of the background noise that we, for example, saw in the early talks in the morning. When they're off, there's kind of also some noise, and
it really depends on which system you're looking at. So BrainScales 2 is not the first system, first neuromorphic chip. So the first actually was spiky. I've got this here with me. I've never used this. I'm too young for this. And so BrainScales 1, which is still operating, this has a lot of noise on the membrane. Here, on this new iteration, you see that the voltage, there is some noise as you zoom in, or at some points you see this, but the noise of these voltages, there's like, you can basically ignore this for there. If you do, for example, the sampling, then it's really important to incorporate this noise in your computations, in your idea of the network that you're doing. So there it's interesting to look at it again.
Okay, so thank you again, Julian.